evening, everyone, and thanks once more for joining us in the Oxford University Museum of Natural History online lecture theatre. We have people joining us from all around the world. Good morning, good afternoon, and good evening, depending on where you are. I can see we've got friends joining us from Russia, from Brussels, from America, and from the Ukraine. So welcome to everyone joining us. My name is Jack Matthews, and I will be chairing tonight's lecture. Just a few reminders about how our webinar Jam online system works. If you have any problems with your connection at any point, do click the reconnect button at the top of the page. Should be able to see that now. That will solve most of the connection problems with the server. Also, you can see we have a chat on the right-hand side. If you click the chat, you can let people know who you are and where you're joining us from today. And it's also a great place to ask any questions you have of our speaker tonight. If you can just mark those by clicking the little circle to the right-hand side, if you're asking a question, we'll curate all of those together and we'll ask our speaker as many as we possibly can at the end of the talk. Just a reminder that all of our lectures are recorded and they go onto YouTube afterwards, our OUMNH YouTube channel. I'll be popping up a link for that in a moment. So if you've enjoyed tonight's talk, do feel free to share it with friends and family. And on that point, we will move to our very special guest tonight, who is Professor Xiao, who joins us all the way from the United States of America because he is Professor of Geobiology in the Department of Geosciences at Virginia Tech. He did his bachelor's and master's at Peking University before going to Harvard for his PhD, and since then has been integrating paleobiological, sedimentological, stratigraphic, and geochemical data to answer questions about Precambrian paleobiology and paleoenvironments, which is what we're very much looking forward to hearing him speak about tonight on the topic of first animals. How did they move? So hopefully, Xiao is going to appear on screen. There we go. Xiao, how is everything in Virginia Tech?
Everything's good. Can you hear me? Is the audio and the video working?
Working perfectly. And so at that point, I will call up Xiao's presentation. First animals, how did they move? I think this is going to be a real treat. Take it away, Xiao.
Okay, thank you very much. And thank you all for coming to the talk from around the world, and particularly those from Asia. It is midnight now, so I appreciate that. So I would like to talk about the motility of some of the earliest animals. But before I do that, I would like to acknowledge my students, my colleagues, and particularly those from the Nanjing Institute of Geology and Paleontology for many years of collaboration. This is not an exhaustive list here. There are many who are not in those pictures, but have contributed to the talk, to the research that I've been doing in the past decade. So I would like to start by sharing this map. It's a map showing the field site where I have visited over the past decade, 2 decades. A very important part of my research is to do field work. And one of the most favorite field site is in Tixi or near Tixi in northern Siberia. This is the upper right pane on the map. This is a small town in the Arctic coast. So here's a picture of our field crew, including Jay Kaufman, Dima Gratz-Duncan, and Natasha Bikova, and a few others from Russian and the United States. It's a very fun field trip, but also it has its challenges. And one of the biggest challenges in doing field trip in Siberia is the mosquitoes. And you can see some of the mosquitoes on my coat, on my field coat. Some of them are really big. For example, this one here, and a few others. We had to always put on our beekeeper's nest to keep the mosquitoes out. It helps somewhat, but it's difficult to keep them completely out because mosquitoes sense heat and CO2. So when we cook, we have the stove and the smoke coming out, and there's always a tornado of mosquitoes swarming around above the stove. And then when you open the lid of the cooking pot, they all dive into whatever you're cooking. In this case, we cooked some onion soup, and the dozens of this very giant mosquitoes end up being a delicacy. But this is not the worst thing about mosquitoes. The worst thing about mosquitoes is that they're swarming in front of you, blocking your view of the rocks. So when I tell field stories like this to my parents, they always ask, why do you choose to go places like northern Siberia or South China or Namibia for that matter? Now I had to explain to them, why I, go to these places, and I always tell them that I was motivated by Darwin's speculation about Precambrian fossils, and particularly Precambrian animal fossils. So in 1859, Charles Darwin wrote, consequently, if his theory about the natural selection be true, it is indisputable that before the lowest Cambrian stratum was deposited, the world swarmed with living creatures. And by that, he meant animal life before the Cambrian. But of course, at Darwin's time, there were no Precambrian animal fossils. But Darwin quickly found a solution. He realized that the fossil record is not complete. So he kept writing. I look at the natural geological record as a history of the world, imperfectly kept. Only here and there, a short chapter has been preserved, and the entire Precambrian chapter is missing, as far as the animal is concerned, from the geological record. So today I want to touch upon a few questions about not only Precambrian animals, but more specifically bilateral animals, and explain in a minute what that means. And I address a few questions. Why animals began to move, when they learned to move, and how they moved when they first evolved. So just to warm up, I'd like to do a little quiz. Don't be nervous. This is more like a vote. So a quiz, you get graded. I will not grade you, but a vote, you grade other people. So you'll probably you'll probably see a sort of poll pop out on your screen, and I give you a few, maybe 10, 20 seconds to fill out the poll. Okay. The question is: the origin of animal motility was driven by the need to A, to find food, B, to find a mate, C, to find oxygen, D, to pursue a prey, and the last, avoid predators. And you can probably see 60 on your screen, 60% of the respondents vote finding food is the main driver, which makes a lot of sense. All animals, they're heterotrophic organisms. They need food. They also need oxygen. They also need to mate. So it really depends on what is the limiting factor, whether food is universally available or whether food is spatially limited and locally concentrated. So go with oxygen and other needs here. So yeah, that's a very good answer, food, and in fact we talk about this in a few minutes. So I mentioned bilaterian, and I want to spend just a few minutes to talk about bilaterian. Bilaterian are probably most of the animals that you're familiar with are bilaterians, from butterflies to bonobos. So here is a family tree of all living animals and their relatives. So all animals in yellow color here. So bilaterians are here. Let me change the color. Bilaterians are this group here, in this family tree here. But animals is more than bilaterians. There are also things like cnidarians here, sea anemone, jellyfish, and corals. So together they're known as eumetazoans. As well as sponge. So this is a sponge, and the ctenophores or comb jellies. So on the right here. So those are the major living groups of animals, and the bilaterians are probably the most capable animal in terms of locomotion or motility. Some sponges can move, but they move very slowly, and their mode of move is really different. The fastest sponge move about a few millimeters a day. And they move by reorganizing all the cells in the body. So kind of like a slime mold type of movement. Certainly very different from earthworms or butterflies or tigers and things like that, or bilaterians. Cnidarians and ctenophores also move a little bit, particularly some of them can swim in a water column. But I would say bilaterians are capable of self-powered directional movement. They're certainly the most effective when it comes to locomotion or motility. So today we're going to focus on bilaterians. Now, there are a lot of extinct animal fossils. So the previous slide only showed living representatives. This slide is a schematic diagram adding the fossils. Once you add the fossils, there's something very important that we need to know, and that is the evolution of animals take many steps to build the animal body plan. And not all animals that ever lived are still with us today. Many of them went extinct. So some of the intermediate steps are represented by extinct animals or animal fossils. So those animal fossils, if they fall outside the living group of animals, so let me draw here. So this is a living group of animal here. If I got an animal fossil that is outside, so this animal fossil that's outside the living group, but it's more closely related to animals than it is living sisters, and that's coronoflagellates, the white branch here. It's still counted as an animal, but to make that distinction, because it doesn't have all the features that collectively define living animals, it has only some. We call this stem group animals or stem group metazoans. Okay, to distinct from the crown group or the minimum clade or the minimum group that include all living animals. So the crown and the stem. So all this are crown, okay, to here, and this is a stem. Together they make the total group animals. This concept of stem versus crown is a nested concept. We have total group and a crown group and a stem group metazoans. And by the same token, we have total crown and stem biolacterians, total crown and stem eumetazoans, and a total crown and stem tetrapods and mammals. Virtually all fossils, all extinct fossils are stem group at one level or another. So with that, I wanna now give you another sort of tool to understand the presentation, and that's a geological time scale. And to illustrate Darwin's speculation about Precambrian fossil record, this is a geological time scale to illustrate that. So the Earth is about 4.56 billion years old, and the geological history of the Earth is divided into the Hadean, Archean, Proterozoic, and Phanerozoic eons. The first rock is about a little more than 4 billion years old. The first fossil, body fossil, some microfossil, is about 3.5 billion years old. The first trilobite, a little more than 500 million years old. Okay, and this is the group of fossil that appeared at Darwin's time abruptly at the base of the Cambrian, Lower Cambrian, that sometimes is described as the sudden appearance of animals in the early Cambrian or the Cambrian explosion. The first dinosaur a little more than 200 million years ago, and all dinosaurs except birds went extinct about 65, 66 million years ago. So the Cambrian explosion, okay, so this is the Cambrian here, it happened very recently in geological time, okay, when you consider the entire history of the Earth. But today we're going to look at this geological time period. I mentioned the Tonian, the Ediacaran, and the Cryogenian and the Ediacaran in the next few slides. So put them in context. The Tonian, Cryogenian, and the Ediacaran all are part of the late Proterozoic. Okay, one way to address Darwin's gap about Precambrian animal history is to use the molecular clock method to estimate the divergence time of animals. And here an example of this molecular clock analysis, and there are many of them. This is just one of the more recent one. So in this diagram, on the top part, what it shows is the family tree of all animals. So this, let me see, start from here. Those are all the animals, living animals, of course. And to study their protein and the genetic sequence and to compare the similarity and the differences, and using a technique called molecular clock dating to estimate the divergence time, basically the branching time of the different animal groups. So the major branching event are all those. So this red branch, and if you project it to the time scale at the bottom, you get a divergence time of about 800 million years, give or take 100 million years. Those come with a very big error bars and uncertainties, but most molecular clock estimate place the divergence of crown group animals in the Tonian period. Okay, but so far there have been no Tonian animal fossils, at least no unequivocal Tonian animal fossils. The oldest hint of animals comes from the Cryogenian period, and is based on not body fossils, but molecular fossils, also known as biomarkers, that is preserved in very old rocks of Cryogenian in age. So this is a geological time on the left here, the Cryogenian, Ediacaran, and the Phanerozoic. This spindle diagram here illustrates the relative abundance of a type of biomarker. And this biomarker is a type of cholesterol So its structure is illustrated here. Okay, it's got a very long name, 24 isopropyl cholesterol. So this is the structure when this molecule is in a live demo sponge. It's a kind of sponge animal. And after the animal died, these lipids, cholesterol, which makes part of the cell membrane of the sponge animal, and is buried and it get degraded and changed over geological time, and it becomes something called 24 isopropyl styrene. So you start from sterol to styrene. Basically, this is what you get in what you pay for in Burger King, and this is what you would get in a cryogenium shale or rocks. So. And some people argue that this molecule is diagnostic of sponge. So the fact that you find this 24 isopropyl cholesterol in cryogenium rocks means that there must be some sponge animals lived in the cryogenium period. But the real body fossils of animals didn't appear until in the Ediacaran period, after 635 many years ago. So this is geological time. Again, the Cryogenian, Ediacaran, and the Cambrian here. The Ediacaran started about 635 many years ago, following the termination of a gigantic, catastrophic glaciation known as the Marinoan snowball earth glaciation. Over the past 50, 60 years, there have been a lot of discovery of Ediacaran fossils, and I'm going to show you a few representative biotas in the Ediacaran period. And one of these biota is the Darwin biota that I've been working on since I was a graduate student many years ago. And here's a fossil on the cover of Nature from the Darwin biota that ranged from 635 to about 550 many years ago. And some of these Darwin fossils have been interpreted as animals or stem group animals or some organisms closely related to animals, but the interpretation is still controversial. And I don't think there's a consensus at the moment. Another representative Ediacaran fossil biota or assemblage is the abalone assemblage. Here is a fossil from the abalone assemblage, a leaf-like fossil. And the abalone assemblage is recorded in Newfoundland in Canada, and it is geological terrain in England. It's about 575 to 560 many years old, and that is followed by the way that fossil from the abalone is about an inch in size. And the abalone is followed by the white sea, perhaps the most important Ediacaran fossil biota, particularly if you are talking about macroscopic fossils. So the white sea is about 560 to 550 many years old, and the best example comes from the White Sea area in Russia and the Ediacaran Hills in South Australia. So the Ediacaran Hill actually gives the name to the Ediacaran period. Here's a fossil from the Ediacaran member in South Australia. This guy seems to have a head and a tail and a left and right side. So this is something very interesting. There's something reminiscent of bilateral animals, which all have a left and right side that are mirror image of each other, and also have a head and a tail, or anterior and a posterior polarization, and a dorsal and ventral polarization. Okay, so those are some of the most important features of living bilaterians. And the White Sea is followed by the Nama assemblage, the last 10 many years of the Ediacaran period. This again is a fossil from the Nama group in Namibia, probably about 545 many years old. And in the Nama assemblage, it's the first time when you see fossils, animal fossils that show evidence of skeletonization and a biomineralization. So this is something that's very important because almost all the fossils you see in the Phanerozoic, they're skeletal fossils. They are biomineralizing skeletons, such as bones and shells and other type of skeletons. And that is followed by then the Cambrian fossil. So here's an anomalocarid fossil from the Berger Shale. One of the things, if you paid attention to these fossils, you probably noticed that as soon as you cross the Ediacaran-Cambrian boundary, you see fossils like this that you're very comfortable, placing in the kingdom of animal, and perhaps in some of the phylum in animals. Whereas all the Ediacaran, quote unquote, animal fossils, it's either controversial as to whether they are animals, or even if you believe that they're animals, it's very difficult to place them in any existing or living animal phyla. So that's a very, very important distinction. So I think there's still something very important happened at the Precambrian boundary, or the Ediacaran-Cambrian boundary. So this diagram from Simon Derrick in 2018 summarized the diversity of Ediacaran fossils, and focusing on the later part of the Ediacaran period. So from 570 to about 540 many years ago. So the three assemblage, the Avalon, the White Sea, and the Nama, they're shown here. Here's some examples of Ediacara type fossils. Okay, most of them are leafy-like. Some of them are sort of discoidal fossils. Some of them are T-shaped. Some of them seem to have a left and right side and a head and tail. And there's still a lot of very bizarre, this guy, for example, 5-foot symmetry. There are 3-foot symmetry representatives among the Ediacara without an N. Okay, so that's different from Ediacaran, which is the Ediacaran period. Ediacara is here we use Ediacara to refer to a special group of macroscopic fossils, such as those ones. Okay, most of them do not move. Most of them are sessile. Some of them have very limited mobility. We're going to talk about a few of them. But I want to draw your attention to this this diagram here that shows this beige color. There's a histogram, the bars, shows the diversity of the Ediacara type fossils. And there's an increase from the Avalon to the White Sea, followed by a decrease and a complete disappearance of the Ediacara biota in the Nama group, and almost all of them disappeared by the early Cambrian. Now that's very interesting. So you kind of see something, the birth and the death of something. Right? This is very different from what the trace fossil record shows. So the trace fossil record here, this is to the right, the purple color. Again, the diversity or number of genera, the same thing here, number of genera of trace fossils from the Avalon, White Sea, and the Nama. And you can see this consistent rise in diversity of trace fossils from Avalon all the way to the Cambrian. So this is very different trajectory than the Ediacara type fossil. Now, obviously, all the trace fossils are made by mobile animals, and mostly by mobile bilaterian animals. So why the dichotomy? There are a few hypotheses, few models explaining this, and in one model, it has been proposed that it's a biotic replacement model. In other words, these mobile bilaterians diversify at the Ediacaran-Cambrian boundary at the expense of the sessile or mostly sessile Ediacara biota, either through direct competition, so they're basically outcompeted, or through some sort of ecological engineering or ecosystem engineering. So it's sort of collateral victim of the divergence or the diversification of mobile bilaterians. An alternative model suggests that is the differential response to an extinction event. So there was an extinction event happened in the Nama assemblage and at the Ediacaran-Cambrian boundary that wiped out most of the Ediacara biota. But for some reason, these mobile bilaterians fared better and suffered a little bit, but then recovered and sustained a renewed diversification after the Ediacaran-Cambrian boundary, and then resulted in the Cambrian explosion after that. Now, to elaborate the biotic replacement model a little bit more, Graham Butt and Soren Jensen proposed this hypothesis called the savanna hypotheses. Basically, what they're saying, and remember we did this poll, finding food, finding mate, and finding oxygen. But basically, what they said is there's more or less sessile Ediacara type organisms are served as a localized food particle, a food source for the mobile animals. So this is basically a driving factor in an environment that food resources is very limited. Just like modern savanna environment, the animal has to hop from one hotspot to another hotspot, and they can only do that if they can move. And perhaps same thing happened in the late Ediacaran that drive the expansion of mobile bilaterians, and including this bilaterian browsers, grazers, predators, and scavengers. In any case, they have to go from point A to point B to find food. Okay, and one can make a similar argument because oxygen was also a scarce commodity in the Ediacaran period. Unlike today, we have a lot of oxygen available. We don't never have to worry about oxygen. But back in the Ediacaran, the oxygen level was a small fraction of what we have today. And not only that, the distribution of oxygen in the ocean was not even and not homogeneous. So oxygen is spatial heterogeneous, and is temporal dynamic. It changes perhaps from day to night, and from shallow water to deep water. So animals have to constantly find oxygens back in the find the oxygen oasis back in the Ediacaran time. Now, the evidence to support or to test this kind of hypothesis has to come from the fossil record. Is there evidence for mobile bilaterians? And is there evidence for bilaterian Ediacaran ecological interactions in the fossil record? Now, I want to quickly review some of the fossil record from the Ediacaran period, particularly focusing on animal motility. And one of the oldest trace fossil is this one from the Avalon assemblage, about 565 million years old, from Newfoundland, the Mistaken Point Formation in Newfoundland. So this here we are looking at this very straight trace fossils. Here's a close up. It's got some of the structures in it, and this has been interpreted as a trail made by some animal look like or resembling sea anemone. In other words, a cnidarian trace. It's not a bilaterian trace. And this is perhaps the only trace fossil from the Avalon. No, to my knowledge, there's no convincing bilaterian animal trace from the Avalon. But once you get into the White Sea assemblage, once you cross into the 560, 550 time interval, you get a bunch of trace fossils, and I'm going to illustrate a few of them. This is a bunch of footprints. So here's one here that has these features that are characteristic of a Ediacaran fossil called Dickinsonia. And here's another one. Okay, and you can almost see some of the very vague structure. And here is another one. Okay, and you get to see this type of structure. This has been interpreted as a series of footprint made by the same individual of Dickinsonia animal, which, by the way, have some sort of left and right and a head and tail differentiation. Not necessary to say it is a crown group bilaterium, but it's certainly a eumetazoan and perhaps a stem group bilaterium. So those are the footprint made by the same animal. That means the animal can move at least intermittently and perhaps passively. It might have some self-powered locomotion, but it's not the continuous movement that you see in worms and other bilaterians. Another example, similar example of mobile animals from the Ediacaran, from the White Sea interval, is this thing called Yogia. It's very similar to Dickinsonia, at least broadly. The central axis here, you got some left and right differentiation. I'm sorry, left and right sides, and maybe a head and a tail differentiation. Okay. And again, it's got several of this footprint behind it. Okay, here's this one. And here's another example, only the tail part of that animal is preserved and a bunch of footprint. So when the trace or the footprint and the trace maker are preserved together, that means the traces are made immediately before death. Okay, so this kind of trace is sometimes known as mortecnia or death traces. So this is interpreted as a footprint, and I think the next, the animation, you can let me see, shows an interpretation of Yogia. So this is a Yogia animal with the reconstruction here. And is soft-bodied. It has no skeletons, no bones, and there are at least a full footprint behind it. The animal probably started from here, quote unquote, jumped from one place to another place, and then died and preserved with its footprint. Okay, and again, this kind of movement is sort of intermittent movement, and but it's a degree of motility among some Ediacaran type fossils, perhaps some stem group bilaterial animals. This fossil also from the White Sea assemblage, Kimborella, it certainly has a left and right side and a head and a tail. And most likely also a dorsal and ventral side, or a back and a belly side. A lot of times this Kimborella fossil is found together with this scratch marks. And you can see many of them here. Okay, this has been taken as evidence for not only moving but also grazing of microbial mat. Kimborella is widely accepted as a bilaterian animal, and it was capable of active, self-powered, continuous directional motility and grazing in this case. Okay? And here again is another animation showing the reconstruction of Kimberella. It has a proboscis-like structure at the head, at anterior end. It probably have a pair of teeth within the proboscis, and they just scrape the microbial scum and collect food particles, and the feed itself, and that make this scratch mark on the microbial mat that is preserved together with the animal. Again, another Mortechnium. But perhaps the most abundant evidence for mobile bilaterians comes from this trace fossil that are very common in the White Sea assemblage. There's something called Helminthoidignites that burrows into the sediment, perhaps beneath a very shallow microbial mat. Again, this is very continuous, and it's self-powered and directional mortality by some animals, although in this case we don't know what kind of animals, although recently it has been suggested that the trace maker of Helminthoidignites have been identified. It is a recent paper published by Scott Evans from Smithsonian in PNAS. So with that, I want to take you to the Yangtze Gorge area in South China to look at the last 10 million years of the Ediacaran period. In other words, the Nama assemblage. So this is a beautiful picture of the Yangtze Gorge. The fossils I'm going to talk about comes from the Shibantan member. Okay, it's made of very thin bedded stinky limestone or bituminous limestone. So when you hammer it, it really smells really bad. And its age is constrained by two radiometric ages between 551 and 543 million years old. So it's definitely part of the Nama assemblages. So one of the first fossils coming from discovered from the Shibantan member is this thing called Parachonia daniensis. So it was found in the 1970s and it was published in English literature in 1986 by Song Weigor. If you can see this, this is a fossil that is better preserved on this side, and you can see some segments here, and it's better seen here also. But it's not until 30 years later when the next Ediacaran type fossil was found in the Shibantan member. And the reason it takes so long for the next specimen to show up is that Parachonia daniensis, as well as most other fossils from the Shibantan, mostly in 1980s, 1990s, mostly come from people's roof. Like I said, the Shibantan member is made of thin bed limestone. They smells, but they split very well. So you can split them to make roof tiles to roof your house. So when we went there, we oftentimes take a pair of binoculars, so looking at people's house or looking at the roofs of their houses. And you can see this roof is failing. The gutter is falling, the roof is leaking, and the owner decided to replace the roof. And he took down all the roof tiles and piled on the ground. And one of my colleague from Nanjing, Charmin Zhou and Chen, they spotted this specimen. Something probably a fragment of pteridinium from the pile, and asked the owner of the house to keep all the roof tiles. And amazingly, they discovered the counterpart from the same roof. That's very amazing. Not only that, we were able to trace, talking to the farmer, we were able to trace where this roof tile was quarried. And we were able to identify or to find the fossiliferous horizon, and it began more systematic excavation of the Shibantan membrane. And over the last 10 years, almost 10 years of excavation, we have recovered thousands of specimens, many of them still in the process of studying, or being studied. Here's a few of them. So a lot of them are discoidal fossils. This is perhaps the most abundant fossil, along with the tubular fossils shown on the right here. So this would be something known as a Himalora that has a disc and this mustache-like appendages coming out of the disc. They're probably holdfast or the rooting structure of some other leaf-like Ediacara type fossils. But there are a lot of leaf-type Ediacara fossils, like this arborea. This is a little tiny thing, maybe a ½ inch in size here. And oftentimes they are oriented by water current. Okay, so they probably felt by water current and oriented by water current. And sometimes on the single bedding surface, you see a handful of those. And here is another specimen, probably a Rangiomorph, a Rangia fossil. There are also possible Dickinsonia. There's also Charnia and perhaps Trapecia. A number of those from the Shibantan member. But what makes a Shibantan member most important is not this body fossil, but the trace fossils, and they can be really abundant. In some fossiliferous horizon, the trace fossils cover 30–40% of the bedding surface. For example, this slab here. And you can take a closer look at the density of these trace fossils. So this trace is probably a burial, because you can see the trace can be preserved as both positive relief and a negative relief on the same bedding surface. It's got this sort of spiral structure on the trace. This is something called probably called stratigraphic, and is a barrel perhaps made by an animal that spinning inside the sediment or in a microbial mat. We still don't know how it works, but we think that it's probably a barrel rather than a trace. This fossil, the trace fossil, is also found in Namibia and featured in the Oxford University first animal exhibit here. But different from our interpretation, this is depicted as a trail on the sediment surface. So in our interpretation, it's not a apifauna animal moving on the sediment surface, but actually an infauna animal moving inside the sediment, not very deep, but inside the sediment or inside a microbial mat. This painting is really interesting because it also shows that some of the trace began to sort of approaching this sessile Ediacara type organism. I don't know whether this is accidental or ecological. Maybe, like the savanna hypothesis, is going to get food. So it's very interesting that in the Shibantan, here's another specimen of this spiral trace, okay, stratigraphic, and it's sort of approaching from different directions to this discoidal fossil, something we call Aspidella or Aenaria, depending on which name you prefer. If this is shown to be ecological rather than accidental, this would indicate some interaction, some ecological interaction between the holdfast or the rooting structure of a sessile or immobile Ediacara type organism and a mobile, most likely bilateral animal. And this would provide some intriguing evidence for the savanna hypothesis and the biotic replacement model. a lot of these Shibantan fossils, trace fossils, they're compound trace fossils, meaning that each individual fossil specimen exhibit several types of animal behavior. Here's one example of those. So in this example, you see a little discoidal structure here. That's probably a resting trace by an animal. And then this is connected with a trail. And some other examples is further connected with a barrel. So you have a barrel moving inside the sediment. You have a trail moving on top, on the surface of sediment. You have a resting trace. Here again, a trace, and followed by a trail or barrel. So this is a compound trace fossil that tells us a lot about the behavior ecology of these trace makers. So certainly a bilaterian animal. Here's another trace, a compound trace. In this case, we're looking at a barrel here, okay, moving within the sediment, and two tracks. This is a track, and also this track. Okay, so based on the cross-cutting relationship, we can reconstruct the animal probably move from here, and then make a U-turn, come back, over cross its barrel, and end up here. And this type of track, so it's got this impressions probably made by an animal with paired appendages or some sort of legs, like modern arthropods and annelids. Here again is an animation to show our interpretation of this trace fossil. We didn't see the buck, so it may be a buck or maybe not a buck, but it's probably something with a paired appendages. Walked on the sediment surface, went into the sediment surface, come out again, and making this sort of shape of love or sign of love trace fossil. Okay. Now, in all these Shibantan trace fossils, none of them are preserved with the trace maker. So that makes it very difficult to say anything definitively about the trace maker. Whether they are segmented, whether they have legs, we can infer, but we don't have direct evidence for appendages and segmentation for that matter. Until we find a motility of this animal called Elengia speciformis. So this is a body fossil with segmentation, so it's very clear. So this is the head, this is the tail, and on the left side, this is a reconstruction with the segments with trilobes. So it's a central lobe in the middle and the two lateral lobes, okay, on the side. So this is a reconstruction on the lower right. And one of the specimens that we found, we have the tail end of that animal, the body fossil, but behind that, it's got a trail behind it. And it kind of have this very vague impression of this body segmentation. So our interpretation is that this part here is a motility, a death march trail, and this is the animal that died while it was making the trace. This is a reconstruction on the in the lower side, and I think the next we can see a animation to show our interpretation. In this case, this is we start with a body fossil, and it is interpretation with the segmentation and the trilobed. Perhaps with some sort of appendage or appendage-like structure or protrusions. Take a pause here and make an impression on the trail. Kept it going, and make another trail, and continue going, and then died. And with the trail behind it preserved together with the body fossil. And this is what we found in these two slabs put together. Okay? So now we can link the trail or the trace fossil and the trace maker together. That gives a lot more information about the animals and how it behaved. And lastly, I want to spend just a few seconds to talk about this very interesting trace fossil. It's made of a series of linearly aligned segments of barrels. Okay, there's 6 or 7 of them that seem to make a very, very nicely aligned. And then we can cut thin sections because we have both the part and the counterpart is oriented, so we know which side is up, which side is down. So basically, this is a transverse cross section. This is up. When you cut something like this with the barrel is missing, all you get is a concavity on the lower side of the bed. This is a top side where you have the segment. So this would be something like this, transverse cross section, and this longitudinal cross section. So this would be something like this cross section. But because we have both the part and the counterpart, we can sacrifice some of the specimens and make thin sections, and to demonstrate that the trace fossil is always associated with microbial mat. So this organic rich laminae, they're just a amalgamation of microbial mat. And like all the trace fossils that I showed from the Shibantan, they're always associated with microbial mat. And we think there's probably oxygen-producing cyanobacteria mat. And since we have both the part and the counterpart, we were able to exclude the possibility that the segment will sort of undulating traces that are made within the sediment. So the gray is sediment, the blue is seawater. If this is the case, then we should be able to piece them together and see the complete undulating trace when the part and the counterpart are brought together. But this is not the case. What we found is that there's some missing segments, a missing part between the segments. The only way to produce this is an animal moving in and out of the sediment, into the sediment and into the seawater. So no trace in this part, and there's a trace, no trace in this part, there's another trace. So when you have the next wave of sediment coming in, there's no barrels within the counterpart in the gaps. So there's nothing here and nothing here. So we were able to conclude this animal moved between the water sediment interface, in and out of the water sediment. And because is the very shallowest sediment is represented by the microbial mat, we infer that the animal moved into the microbial mat, and not just microbial mat, it's live microbial mat. And it probably explored either oxygen or perhaps food when it visited the microbial mat. So this is very interesting. This is another line of evidence that suggests that the animals interacted, not only mobile animals, but they interacted with microbial mat, with live microbial mat. And if we can show that this is related to oxygen mining, then it's an important piece of evidence suggesting that animal might have that they're limited by the availability of oxygens, and they have to find the oxygen oasis. And remember, microbial mat, within microbial mat, the oxygen concentration fluctuates strongly between day and night, because during the day they photosynthesize and produce a lot of oxygen. During night, they use up all the oxygen, and the microbial mat becomes inhabitable and lethal to many animals. So I think I'm going to, here's another, the last animation. If you can, Jack can show the animation very quickly, just 20 seconds, I think. Without interpretation, in this case, we don't know the trace maker, what the trace maker look like. So the red worm-like animal is just a made-up animal here, but this is the segments of barrels. The animal come in and out. Then it preserved this 7 or 8 segments of barrels in the Shibantan. So I think I'm going to end my talk by, just thinking about this. So as we are working on the Shibantan fossils, we can't help just wondering, what Darwin would say if Darwin had seen these fossils, right? I think he would definitely be pleased to see any fossils, any animal fossils from the Precambrian, because he predicted that there would be Precambrian fossils, but there were no Precambrian fossils back in his time. And I think he would also very pleased to see that the complexity, motility, bilaterians, segmentation, appendages, interaction with microbial mats, interaction with sessile Ediacara type organisms. So he would be amazed to see not only the diversity but also the complexity of these Ediacaran animals. And I think he would strongly encourage us to look closer, look harder, and look deeper in the geological record, because in his mind, Darwin thinks that animal had a much deeper history than the Ediacaran. And so did all the molecular clock analysis. So I think I would like to invite all you to join us to study Ediacaran paleobiology and the Precambrian paleobiology in general. Maybe you will make the next most significant discovery. Thank you very much.
And there you go, everyone, a call to arms. You could be the next great fossil discoverer. Thank you so much to Xiao for a wonderful talk at some absolutely fascinating fossil discoveries and the wonderful interpretation and animation that brings that to life. Before we get to some questions, and a reminder, do pop your questions in the chat if you have any questions, and we will get to those in a moment. Just a quick reminder on what is coming up in our series. In 2 weeks' time, we are very pleased to have Professor Derek Briggs from Yale speaking to us. He'll be discussing the Cambrian explosion, and I think we'll be talking a bit about the Burgess Shale as well. Then 2 weeks after that, on September the 9th, we'll be going up into Canada, the University of Saskatchewan, with Professor Gabriella Mangano. And then finishing off the First Animals series within our online lectures and talks, we'll be hearing from Professor Rachel Wood from the University of Edinburgh on September the 23rd. As always, we like to hear your thoughts and opinions. If this lecture has changed your opinion on the evolutionary origin of animals or added to your understanding of the topic, why not add why in the chat? We always love to hear from you about these things. And also, if you're able to, please do consider donating to the museum. The museum is a charity. We obviously run our collections, but also the building itself, which will be hopefully opening soon. And also our collections, our educational work and things like this. So please do consider donating. The link is up in the chat at the moment. And at that point, I think we will get to some questions. And I think to start off, we'll go to Kaylee Mills, who asks, in regard to the change in diversity through the Ediacaran and through the Cambrian, and that increasing diversity, she asks, how much of that change in diversity is a relic of the number of Lagerstätten of those ages?
Yeah, I think to some extent the diversity certainly is biased by the availability of Lagerstätten. Lagerstätten is just deposit with exceptional preservation of fossils. Particularly given that all the Precambrian, most of the Precambrian fossils, with the exception of a handful of them, are soft-bodied organisms. So you require certain very special kind of preservational environment to capture them in a fossil record. And the same can be said in the early Cambrian. Although you get a lot more skeletal fossils in the early Cambrian, the best preserved fossil, early Cambrian fossils, and the middle Cambrian fossils, like the Xiao, are probably soft-bodied fossils. That said, I think there is some truth to the diversity pattern that we saw. Certainly for the trace fossil record, is less prone to preservational bias. So the trace fossil record is I think more reliable than the body fossil record. Now the body fossil record, if we just focus on the Ediacaran fossils, I think we can also say with some confidence that the decline in the diversity of Ediacaran fossils during the Nama assemblage, in other words, the last 10 million years of the Ediacaran period, is probably real than a artifact. And the reason is that if you look at the number of Lagerstätten, there are actually comparable number of Nama assemblage Lagerstättens. So far, we have, for example, Shibantan, the Nama group, the Kalispit in Siberia, and the Corumba in Brazil. So they are a reasonably good number of assemblages, but all of them have very low assemblage diversity than either the Ediacaran Hill, Ediacaran Membrane in South Australia, or the White Sea. So if we have 8 or 9 or 10 Nama assemblages, and none of them have a what we call alpha diversity, than the White Sea and Ediacaran, I think the chances that there's probably a low diversity in the Nama than in the White Sea. But we cannot completely rule out that taphonomic bias played a role. But it's just Mark Laflamme and others have wrote several papers to discuss this issue, and including Drew Masente. So I can give you these references if you are interested. They discuss these issues in great length in their papers.
Now, for the next question, we will go across to Newfoundland, a place very close to my heart. And we've got a few questions about frondose fossils for everyone at home. Just a reminder, a frondose fossil looks something like this. This is a Charnia, which you might remember from Frankie Dunn's talk a few weeks ago. And Morrison asks, is it thought that the frondose Ediacarans hopped along the seafloor like anemones? So were those frondose Ediacaran organisms, were they moving around?
Interesting idea. In principle, I think it's theoretically possible, but the real question is whether there's any evidence for that. Now, there are a lot of frondose Ediacaran fossils in Newfoundland. And the only trace fossil from Newfoundland is the trace that Alex Liu described, and he interpreted this trace fossils as a trail by some Cnidarian sea anemone-like animals. Whether that is a frondose Ediacaran fossil or not, I would love to see evidence for that. Many of these frondose Ediacaran fossils have a discoidal rooting structure or holdfast, and in some of the Newfoundland surface, you have thousands of these holdfasts. I think it would be interesting to look at this very closely to see whether there's any evidence for movement of this holdfast. From what I can tell, a lot of these holdfasts are buried in the sediment, so they're not just on the surface of the sediment. So that make it a little harder for them to move like a sea anemone. But that's just my hunch. A short answer to your question is that there's no direct evidence for that, but it is theoretically possible.
And while we're on frondose fossils, we'll go to a question from Phil in Yorkshire, who, and we'll put some extra explaining around this. Phil asks, with the Shibantan arborea specimens, were they felled by turbidites similar to those found in Newfoundland, or does the carbonate sediment composition indicate a different mode of burial? So for everyone at home, the arboreas are another frondose fossil, and what Phil is asking is, was there a marine avalanche of sediment that pushed these over before they were preserved, or is there something else going on with the carbonates?
Very good question. Very good question. I didn't have time to go into any detail about the preservation. I think you're certainly right that the turbidite or some sort of sediment flow played a very key role in the preservation of these arborea fossils. And in fact, the Shibantan was deposited below the fair weather wave base, but above the storm weather wave base. And there are a lot of those little tiny microturbidites in the Shibantan membrane. And oftentimes, when you have the Ediacaran fossils just above a microbial mat, but just below a sort of sandy packstone or grainstone. There's technical terms, but basically a wave of sandy material coming in. So I think it's plausible that those arboreal.
It looks like we're having a little bit of an issue with the connection with Shuhai. While he hopefully reconnects, just a reminder that our next talk is by Derek Briggs, and the link should be up there on the right-hand side. Hopefully we haven't completely lost Shuhai. The link should be up to Derek Briggs's talk where you can register. If, of course, you've enjoyed this evening, you can watch it all again on YouTube, on the OUMNH videos YouTube channel. And, of course, do feel free to share this with friends and family who might be interested in watching this or other lectures again, or indeed get them involved in the ones that are coming up. Derek Briggs in 2 weeks' time, followed 2 weeks after that by Gabriella Mangano. And then 2 weeks after that, we have Rachel Wood from the University of Edinburgh. It looks like Shuhai is just reloading. We have a few more questions. Hopefully, if he can get back, we will ask a few questions. And thank you for all your responses so far in the chat about how you've been finding this of interest. We have a few more questions. Hopefully, we'll get to that in a moment. While we are waiting, thank you for everyone who took part in the poll earlier. I can also share two links. We had, if you were intrigued by our fossil model here, this is printed on a 3D printer. These were virtual models that were created for our first animals exhibition, which is available on the website. Even though it's not open to the public at the moment, you can log in and experience it virtually. And also, if you go onto the museum's Sketchfab account, which you should be able to see in the chat now, you will be able to see these models. You can play around with them in augmented reality on your phone, and you can also download them now. So if you have a 3D printer at home, you too can print your very own Charnia or some other fossils that have come up in recent talks. You could print your own Heutia. And I can see that Shuhai is back in the room. And I can hear Shuhai.
Yeah, my internet went off, so now.
Apologies, Shuhai. We're still ready to go. If you pop your camera back on, we're hopefully we can do a few more, just a few more questions. Thank you for joining us back again.
Yeah. But to complete, the turbidite certainly played a role, but also very early lithification, and it probably fueled or facilitated by the degradation of this buried microbial mat. That the degradation provide some sort of localized environment that facilitate calcite precipitation. Played a very important role also.
Excellent. Apologies for that connection issue. We will move on with the questions. We have two really great questions from Peter Holland. The first one of which he asks, I'm curious, was the owner of the roof tiles interested to hear about the science when you explained your interest in the fossils?
Yeah, certainly. Yeah, they're very excited, and they helped us immensely in the excavation because this is a very labor-intensive excavation, and we cannot spend a lot of time in the field. And the owner and his friends in the village helped us to excavate this site. Unfortunately, they don't speak English, so they won't be able to understand our discussion here. But we do explain them the scientific importance of these fossils. And now, if you go visit this area, there are murals of Ediacaran fossils in the village, and people paint these Ediacaran fossils on their walls. And they learn about these Ediacaran fossils in their life, more than 500 million years old. And some of them have developed very special skills to identify these Ediacaran fossils. And sometimes they're better than us in spotting and recognizing the Shibantan fossils. This is amazing.
Another wonderful example of how local knowledge is so important when finding these wonderful fossil deposits. And the second part of Peter Holland's question was asking that a generalization is often made that the Precambrian-Cambrian boundary is marked by a transition from a primarily 2D microbial mat-dominated world to a 3D world. And do you think that generalized view is challenged by your finding these complicated trace fossils in the Precambrian?
To some degree, I still think the Ediacaran-Cambrian boundary is an important geological and evolutionary divide. But the simplistic view of Cambrian fossils, trace fossils, or animals just on the surface and never dig into the sediment, I think need to be modified. Not only our data, but others. For example, I think there's a talk by Gabriela Mangano, and they will talk about some of the complex Ediacaran trace fossils from Namibia, for example. So I think there's slight modification and enrichment with the enrichment of Ediacaran paleontological knowledge. The Ediacaran animal diversity, animal behavior is more complex than what it was thought previously.
Yes, so if you want to find out more about trace fossils, do tune in again in 4 weeks' time for Gabriela Mangano's talk from the University of Saskatchewan. And our final question tonight goes to Nishi, who asks, how can we rule out the movement by water of these organisms? How do we know that they were actively mobile rather than being pushed around by their environment?
Another really important and interesting question. And for some of those, I think it's difficult to rule out this possibility. For example, maybe Dickinsonia. I think water current may play a role in lifting this animal and then dropping it off, and perhaps Yogia. But water current is, at least water current alone, is difficult to explain the burrows, the movement within the sediment and microbial mat. So tunneling in the sediment, you can't do it with only water current. You need to have some sort of muscles or hydroskeletons and perhaps appendages to help you maneuver within the sediment.
Excellent. Thank you so much once again, Shuhai. This has been an absolutely fascinating talk and really well brought to life by all those wonderful animations and the wonderful, wonderful fossil discoveries. So thank you from everyone attending tonight for joining us from across the pond. Thank you to everyone who's joined us tonight to tune in. Hopefully we will see you again in 2 weeks' time for Professor Briggs's talk. And in the meantime, stay safe, and we will hopefully see you soon. Thank you once more.
Thank you very much. Thanks, Jack. Goodbye.